Let's extend our discussion on DNA damage. We already learned about the types of DNA mutation from our discussion of the genetic code. So we needed to understand what these mutations are so that we appreciate why the genetic code is redundant or degenerate. So now we will be discussing the causes of DNA damage. The nitrogenous bases in your nucleotides they possess a certain degree of reactivity, and more so because they contain pi electrons, right? And because of this, even without enzymes, some of your nitrogenous bases can actually undergo certain transformations. However, these transformations occur very slowly. And since your cell has its own repair mechanisms, then this is nothing to worry about. However, if there are certain chemical reagents or factors that hasten these transformations, and their rate is something which your repair mechanisms cannot keep up with, then that's something to cause concern. So, aside from non enzymatic transformations, we also have external agents that cause DNA damage. So, let's take a look first at non enzymatic transformations. First is spontaneous deamination. So, your nitrogenous bases can undergo this process. And we can see four examples here. In particular, we will be focusing on the deamination of your cytosine. So your cytosine is deaminated, and this is transformed to a carbonyl functional group, and this is actually uracil. Now recall that it was mentioned that there is a reason why you don't have uracil in your DNA molecule. That is because of this spontaneous deamination of cytosine. Your cell is able to repair this transformation. It can revert back the transformed uracil to the original cytosine. So there is a mechanism for that. The thing is, if your DNA molecule already has inherent uracil in it, the mechanism will not be able to distinguish the uracil that was transformed from the deamination of cytosine and the original uracil that is already there in your DNA molecule. Hence, nature saw to it that there won't be any uracils in any DNA molecule so that the integrity of this genetic molecule will be intact. By the way, the rate by which cytosine is spontaneously deaminated and becomes uracil is 1 in 10 million. So that would mean 100 spontaneous transformations in 1 day or 24 hours. So the repair mechanism can keep up with that. So it's not something that you need to worry about. Next is lesion formation. So when we say lesion, this term actually this refers more to the physical consequence. So it's the consequence of the transformation to the physical structure of your DNA molecule. That's what we mean by lesion. But to be more specific, this lesion is brought about by nitrogenous bases that are removed from the nucleotide. So we can call it an apurinic site or an apyrimidinic site. So you don't have a purine or you don't have a pyrimidine at that specific site in your DNA molecule, or generally we can call it a basic site. So for example, your guanosine residue in a DNA molecule can be transformed spontaneously, and this requires water. The process is a hydrolysis reaction, which cleaves this glycosidic bond. To be more specific, this is an N glycosidic bond. So glycosidic because it's linking the molecule to a sugar or a carbohydrate, and it's N because the linkage is via a nitrogen atom. We can also have an O glycosidic bond via oxygen. So this is an N glycosidic bond, which is destroyed in this transformation, removing the purine. Next is the formation of pyrimidine dimer. So this is now, we are now starting DNA damage by external agents. So the formation of pyrimidine dimers is triggered by the exposure to UV light or radiation. So the energy from your UV light or radiation triggers the reaction. So there are two possible outcomes. And take note that um, this will most likely happen for two adjacent thymines. So if you have two thymines in your DNA sequence next to each other, then the likelihood of forming pyrimidine dimers when this gets exposed to UV light or radiation is very high. So what happens here is that the energy from your UV light or radiation initiates a radicalic reaction. So a radical is formed in one of the carbon atoms, and this proceeds via a formation of a cyclobutane between the two thymines. So you can see the cyclobutane there involving two carbon atoms from each thymine. So this also creates a lesion. To be more specific, it leads to a kink in the structure of your DNA due to the formation of the cyclobutane dimer. Aside from the cyclobutane dimer, another consequence of exposure to UV light is the formation of a so-called 6,4 photoproduct. This is also a radicalic reaction, but instead of forming the cyclobutane dimer, what you form is a covalent bond between carbon 6 of one thymine and carbon 4 of the second thymine. This will also cause a kink in the physical structure of your DNA molecule. Next are deaminating agents. So in the previous item, your external agent would be high energy, right? So that high energy can come from UV light or radiation, such as X-rays. Now we have chemical reagents. To be more specific, deaminating agents. So from the term itself, these chemical compounds cause the deamination of your nitrogenous bases. This is different from the spontaneous deamination that we saw a few slides back. So the most commonly understood deaminating agent are nitrous compounds. So we have nitrosamines, sodium nitrite, and sodium nitrate. So these molecules cause deamination. You can actually find nitrous compounds in frozen food products such as sausages, for example, because these nitrites are food preservatives. However, the concentration used as a preservative is very low, so it's not that concerning. And although they can cause deamination of your nitrogenous bases, the cellular repair mechanisms can address that. However, if you eat these frozen uh, products on a regular basis, like every day, for example, then even if each package contains a very small concentration of the nitrite or the nitrous compound, then if you eat it every day, then you're slowly accumulating the preservative, right? So I think it's wise if you moderate the consumption of products that contain these preservatives. Next are alkylating agents. Alkylating agents introduce alkyl groups, commonly methyl groups, to your nitrogenous bases. So here are examples of alkylating agents: dimethyl nitrosamine, dimethyl sulfate, and nitrogen mustard, which is actually a chemical used in chemical warfare. Okay. Here we see S-adenosyl methionine. This is naturally occurring in your cells because some of your nitrogenous bases are actually required to be methylated. So their source of methyl is S-adenosyl methionine. Okay. However, if you consume, or if a lot of these external agents like the dimethyl nitrosamine get inside your cells, then they can cause excessive methylation of your DNA, and we do not want that. So one of the common consequences of these alkylating agents is the methylation of guanine. So the oxygen there at the sixth position is methylated, and this no longer forms the required GC base pair. So the Watson-Crick pair is supposed to be cytosine, right? But instead it pairs with thymine. So instead of the GC base pair, if your guanine gets methylated, it will tend to pair with thymine. And if you do another round of replication, that thymine will now pair with an AT, with an adenine, right? So the original base pair that you have in your DNA was a GC, but because of methylation, this G, which is methylated, will pair with a thymine, and then with another round of DNA replication, that thymine will now pair with an A. So you now introduce a mutation from GC to AT base pair. Next is adduct formation. So the adduct formation is primarily due to benzoapyrines, which is a class of molecules commonly found in cigarette smoke, for example, and also in soot. So smoke. So these benzoapyrines in the presence of oxygen can undergo several transformations. Oxygen and water can undergo several transformations, and then this benzoapyrine can covalently link itself to your nitrogenous base. Okay, like for example, guanine here, you have the amine group there that can act as a nucleophile and can attack the epoxide ring. So that causes the opening of the epoxide here, and forming a covalent link, a permanent covalent bond with the benzoapyrine. And then since part of your benzoapyrine is aromatic, it can intercalate. So it positions itself between the base pair ladder of your DNA, and that is what we call the adduct. So it also changes the physical structure of your DNA molecule. And we have oxidative damage caused by ROS, or reactive oxygen species. We are actually exposed to these ROS on a daily basis. There are reactive oxygen species produced naturally inside your cells, but at a minimum. So they are byproducts of the metabolic reactions taking place inside your cells, and there are enzymes that eliminate them. There are, of course, reactive oxygen species produced by exposure to UV light, and also to the cosmic rays that enter the Earth from outer space. So those energetic radiation can also produce reactive oxygen species inside our cells. But again, we have mechanisms to eliminate them. But if they get excessive, like you get lots of ROS, then your cell will experience what we call oxidative stress. Now, what are examples of these reactive oxygen species? We have peroxides, of course, O22 minus. We can have hydroxide radicals. So it's an OH, but instead of a full negative charge, it has a unpaired electron. It can be an oxygen molecule with an unpaired electron. So these are the typical reactive oxygen species that can be formed inside the cell. And the most common one is the hydroxide radical. So when your cell experiences oxidative stress, this could lead to a lot of damage to your DNA. There are several types of damages that can happen, like the direct oxidation of your nitrogenous bases, as what we've seen in this slide, wherein your guanine gets so it undergoes oxidation at the 8th position, carbon 8. So there you have the carbonyl functional group. So that's your 8-oxoguanine. You can have base mismatches because of these oxidation of your nitrogenous bases. Your reactive oxygen species can introduce single strand breaks. It can cause cross-linking, interstrand cross-linking. Okay, so it can also cause double strand breaks. So all of these, if your DNA becomes overwhelmed by these damages, it can lead to necrosis, cell death, apoptosis, dysfunction, etc. Of course, the organism is already sick. It's in a diseased state. Okay, so with these non-enzymatic transformations and external agents, there is a possibility that DNA mutation can take place. Okay, so when we say DNA mutation, it's basically an alteration in the DNA structure, in the DNA sequence that destroys the blueprint. Okay, because you're essentially changing the DNA sequence. So you are changing the information encoded in the organism's DNA. So DNA mutation can have several causes aside from what we discussed, the non-enzymatic transformations and the external factors. Your DNA polymerase is not perfect. It's not 100% perfect. So while performing replication, it can actually introduce errors, and that can eventually lead to a mutation. Of course, endogenous agents like your ROS that are inherently produced inside your cells, and of course, exogenous agents. So we already mentioned about the types of mutation. So we have transition and transversion mutation when we're talking about base alteration. We have the single nucleotide mutation like point mutation, insertion, deletion. And we also have the types of mutation based on the effect on the phenotype. So is it a silent mutation? So the sequence of your DNA is changed, but the protein product retains its amino acid sequence. And again, this is a consequence of the degeneracy of your genetic code. We have a missense mutation wherein a different amino acid will be incorporated in the protein molecule. This amino acid could be similar to the original amino acid. Let's say instead of the original leucine, isoleucine gets incorporated, and probably that's okay. So the protein molecule will still be able to do its function. But it can also be a drastic change. Like, for example, if you have the originally lysine, which has a positively charged side chain, and then it gets changed to leucine, then that will definitely affect the function of the protein molecule. Then we have a nonsense mutation. Nonsense mutation means your translation process encounters a premature stop codon. So, somewhere along your mRNA transcript, the mutation causes one of your codons to be transformed into a stop codon when supposedly it should call for an amino acid. So, what you get is a premature protein molecule or a shortened gene product, which of course is non functional. So, what are the repair mechanisms used by your cell to address these damage to your DNA? So, we have four types of repair mechanisms. We have mismatch repair, and these are the enzymes involved in this repair system. So, the type of damage addressed are mismatches. Okay, we have the base excision repair. So this repair mechanism particularly addresses the base itself, the nitrogenous base. So from the name of the repair mechanism, what this does essentially is it removes the damaged nitrogenous base and then eventually repairing it with the correct nucleotide. So these are the enzymes involved. Then we have nucleotide excision repair. So this one addresses lesions. Okay, so it removes more than one nucleotide to address the DNA lesion. So it doesn't simply remove a base, a nitrogenous base, but it removes a certain segment of one strand of your DNA. So you have there nucleotide excision repair. Then we have direct repair. So any other type of damage not mentioned is addressed by direct repair, such as your thymine dimers, for example. So thymine dimers can be directly addressed by your so-called DNA photolysases. So we will go through each of these repair mechanisms one by one, starting with mismatch repair. So the mismatch repair actually takes place during DNA replication. So again, your DNA polymerase 3 is not perfect. So sometimes, like 1 in 10 million times, it will introduce a mismatch. So with this mismatch repair system, it increases replication fidelity by a factor of 100 to 1,000. Okay, so since this is taking place simultaneously with DNA replication, it is the DNA polymerase 3 itself that performs the repair. Okay, but you need other enzymes with your DNA polymerase 3. Now, the question is, we know that DNA replication is semi-conservative. So what you have is one strand from the parent DNA and you are synthesizing a new daughter strand. So the result is a double-stranded DNA, right? So how should the cell, if there is a mismatch, the cell should be able to recognize the daughter strand. So it should repair the daughter strand, but not the template strand, which came from the parent DNA. So this is where your DAM methylase comes in. So this is in the context of E. coli. Okay, so the E. coli has a mechanism for it to recognize an old DNA strand and a newly synthesized DNA strand. And in fact, this mechanism is also used by the E. coli to differentiate its own DNA from a foreign DNA, let's say inserted by a virus into the E. coli cell. So the DAM methylase methylates adenine, okay? And to be more specific, so it just doesn't like randomly methylate all of the adenines in the DNA molecule of your E. coli. it has to be an adenine found in the sequence GATC. Okay, so it methylates all of those adenines found in the GATC sequence. And when the E. coli cell starts to divide, so it's undergoing fission, it's reproducing, you have one of the strands methylated, and of course, you are synthesizing a new daughter strand. So this is what we call a hemimethylated DNA because only one of the strands is methylated since the other strand has just been synthesized. So the mismatch repair mechanism takes advantage of this because if your DNA polymerase needs to correct some mismatches, it can recognize the new daughter strand because it's not yet methylated. However, if the replication is complete, after some time, your DAM or your DNA adenine methylase enzyme will now methylate the daughter strand as well, the newly synthesized strand. So if no correction has been made prior to this process, this methylation reaction, then the mismatch is retained in the new copy of DNA. Okay, so the methylation takes place at the carbon 6 position, at the amino group at the carbon 6 position. So how does mismatch repair take place? So again, it needs some proteins such as these mute H, mute L, mute S, and also some nucleases. So let's watch a video first on mismatch repair before going into the details.
This is a video of a 3D animation on the mismatch repair pathway in E. coli. In the cytoplasm is the E. coli's DNA, the molecule that contains all of its genetic information. DNA is made up of nucleotides, each of which contains a phosphate sugar backbone and an organic base. The two strands of DNA are held together by hydrogen bonding between complementary base pairs. Adenine pairs with thymine and cytosine pairs with guanine. DNA mismatches occur during DNA replication. For simplicity, this animation will show DNA as a flat molecule. Before replication can begin, a helicase needs to unwind the DNA molecule. In E. coli, DNA polymerase III catalyzes the incorporation of new complementary bases to the new DNA strand. This is only the alpha and beta units of the DNA polymerase III enzyme. However, it is possible for DNA polymerase III to incorporate a non-complementary base to the new strand. This is known as a mismatch. A mismatch could result in a faulty protein being coded for, so it needs to be repaired. The first step in the mismatch repair pathway in E. coli is recognition of the mismatch by the mute-S homodimer. Mute-S recognizes the mismatch and binds to it. Then, the mute-L homodimer binds to mute-S, ready for a later step in the mismatch repair pathway. Meanwhile, up to a kilobase away, the mute-H protein recognizes a GATC site. Mute-H is a weak endonuclease, which will create a backbone incision between the guanine and adenine residues on the newly synthesized strand when activated. Mute-H is able to differentiate between the newly synthesized strand and the template strand by the lack of methylation markers on the newly synthesized strand. This is why the pathway is also known as methyl-directed mismatch repair. In order for mute-H to become activated, it needs to bind with mute-L. This causes the DNA to bend. The newly formed backbone incision allows an exonuclease to enter and begin excising nucleotides from the newly synthesized strand. The exonuclease excises the mismatched nucleotide and stops excising just beyond it. Now, DNA polymerase III can begin to resynthesize the new strand, inserting the correct complementary base where the mismatch used to be. The final step is for DNA ligase to ligate the backbone nick. The mismatch pathway is now completed, and all the newly synthesized DNA is complementary to the template strand.
So that is how your mismatch repair takes place. So let's take a look at what happens first once a mismatch is made. So the mute S and the mute L will be recruited to the site of your mismatch. So it's the mute S that recognizes the mismatch, and then you have the mute L that also binds to the mute S, which initiates the mismatch repair mechanism. And then the mute H, there are actually two mute H molecules that will bind to your mute S. And this mute H will be the one to scan the DNA strand in both directions for methylated adenine. So what happens is that the DNA is actually looped on your mute L mute S complex. So the two mute H molecules are able to scan both directions. Okay. And then if one of them encounters first a methylated adenine, then that's where it does its function, which is to cleave at the guanine position. So it will cleave at the guanine position, and then all of this complex will disassemble. And the next thing that will happen is that, so once you have the nick there, you now import your nucleases because you need to remove a segment of the DNA surrounding the mismatch so that the DNA polymerase III can redo the polymerization, but this time with the correct base pair. So there are actually two pathways that can happen, and this depends on whether your nick is to the right of the mismatch or to the left of the mismatch. So if our perspective is on the newly synthesized DNA strand, so we are going from 5' to 3', right? From left to right. So this is the 5' end of your daughter strand, and this is the 3'. If it is upstream of the mismatch, then you need exonuclease 7 or the RecJ nuclease to remove an oligonucleotide. Okay, so this oligonucleotide will contain the mismatch base. Okay, and then if your nick is downstream to your mismatch, you need exonuclease 1 or exonuclease X. So the difference between the two mechanisms is that here, your exonuclease 7 or RecJ nuclease is a 5' exonuclease. So it removes nucleotides from the 5' end one by one. So remember, it's an exonuclease. So it removes nucleotides one by one at the 5' end. Meanwhile, your exonuclease 1 and exonuclease X is a 3' exonuclease. So they remove nucleotides one by one from the 3' end. Okay? So for the action of your exonuclease 1 or exonuclease X, it's going from right to left. So they are removing the nucleotides one by one from right to left. But for exonuclease 7 or RecJ nuclease, their action is from left to right. They are removing nucleotides one by one from left to right. Again, if we are going from the 5' to 3', left to right. Okay? And then, as what you saw in the video, once you remove that segment with the mismatch, the DNA polymerase III will do its thing again and resynthesize the oligonucleotide, this time with the correct base pairing. And then your DNA ligase also performs its function of joining the nick. Okay, so that is mismatch repair. Next is base excision repair. So this addresses the repair of abnormal bases such as non-enzymatic due to non-enzymatic transformations. An example we saw was the spontaneous deamination of cytosine to uracil. This can also happen during replication, your base excision repair. So let's watch a video so that we can have a visualization of how base excision repair happens, and then we will go into the details.
Some damaged DNA bases are repaired by a system called base excision repair. Base excision repair works differently from nucleotide excision repair, which clips out a section of DNA containing a pyrimidine dimer or other defect to replace it with new DNA. Base excision repair acts on other kinds of defective DNA. For example, certain bases that are not supposed to be in DNA, such as uracil, are removed by base excision repair. If the base cytosine spontaneously deaminates, it forms uracil. Uracil can also be incorporated erroneously into a strand during DNA synthesis. If uracil forms in the place of cytosine, it would have a mutagenic effect. Whereas cytosine forms a base pair with guanine, uracil forms a base pair with adenine. During DNA synthesis, DNA polymerase would incorporate an incorrect nucleotide into the growing strand. A few other types of damage are also repaired by the base excision repair system. For example, when adenine becomes deaminated, it forms an unnatural base called hypoxanthine. A mutagen can also damage adenine by alkylating it, converting adenine to 3-methyladenine. Normally, adenine forms a base pair with thymine, but hypoxanthine in its place is mutagenic because it forms a base pair with cytosine. 3-methyladenine is even more damaging. When it is encountered in the replication fork, it stalls DNA synthesis. Consequently, repairing these mutations is vital for survival. Base excision repair employs a battery of glycosylase enzymes that can recognize and clip damaged bases from the phosphodiester backbone. Hypoxanthine DNA glycosylase, 3-methyladenine DNA glycosylase, and uracil DNA glycosylase recognize hypoxanthine and 3-methyladenine and uracil respectively when these bases are present in DNA. The glycosylases cleave the bond connecting the damaged base to deoxyribose in the phosphodiester backbone. The result is an intact phosphodiester backbone missing a base. The site is called an AP site. In this case, it is missing a purine and is therefore an apurinic site. If it were missing a pyrimidine, it would be an apyrimidinic site. The next step in base excision repair involves AP endonucleases that specifically cleave the phosphodiester backbone at AP sites. The 3' end produced by the AP endonuclease provides a starting point for DNA polymerase I to start adding nucleotides. At the same time, the polymerase uses its 5' to 3' exonuclease activity to degrade the cleaved strand downstream of the AP site. DNA ligase then seals the remaining nick, and the repair process is complete.
So that is how base excision repair takes place. Okay, so the video was actually enough for the discussion. The only thing you have to remember is that there is a unique enzyme that recognizes any abnormal base in your DNA. And this time, the correction is performed by DNA polymerase 1 instead of DNA polymerase 3. The next repair mechanism is nucleotide excision repair. So this is generally used to repair DNA lesions, which are kinks in the DNA structure. So generally, what happens here is that your xi nuclease cleaves a segment of your strand which contains the lesion. So in E. coli, it cleaves up to 5 bases away from the lesion downstream and up to 8 bases upstream to the lesion. And then the DNA helicase removes this because it needs to unwind, right? So after the cleavage, DNA helicase catalyzes the unwinding so that the oligonucleotide can be removed from the double strand, and then DNA polymerase 1 does its thing of incorporating the correct nucleotides using the template strand, and then DNA ligase joins the nick. In the case of humans, a longer oligonucleotide is removed, so it can cleave up to the sixth nucleotide downstream of the lesion and up to the 22nd nucleotide upstream of the lesion. So it generates a longer oligonucleotide. So it's composed of 29 nucleotides. In the case of E. coli, it's up to 13 nucleotides. But it's the same. You have the counterpart of DNA polymerase 1 in humans or in eukaryotes doing its thing, and then DNA ligase fixing the nick. So here is an animation of nucleotide excision repair.
Nucleotide excision repair is an important general process that cells use to correct many different types of DNA damage, including UV-induced thymine dimers. Although present in all eukaryotes and prokaryotes, the molecular mechanism of this system is best understood in prokaryotes. This animation will illustrate the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called UVRA, UVRB, UVRC, and UVRD. UVR stands for ultraviolet light repair of pyrimidine dimers. But remember that this system is also used to repair other types of DNA damage. In addition, DNA polymerase and DNA ligase are used to complete the repair. The process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of 2 UVRA molecules and 1 UVRB molecule. This trimer moves along the DNA, scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UVRB protein remains at the damaged site, while the 2 UVRA proteins are released. UVRC then binds to the site and makes 2 cuts on the damaged strand. Both cuts are several bases away from the damaged site. UVRD, a helicase, then binds to the site and separates the segment of damaged strand from the rest of the DNA molecule. UVRB, C, and D are then all released from the damaged region. This leaves a gap that is filled with the proper nucleotides by DNA polymerase and then sealed by DNA ligase. The end result is that the damaged DNA has been replaced with the correct bases.
Okay, so that's how nucleotide excision repair is performed.
Nucleotide excision.
Okay, and finally, we have direct repair, which addresses other types of damage. Okay, so these are mechanisms that do not remove a base or a nucleotide, because so far what we have seen, the mechanisms involve removal of at least a base, right, or a nucleotide. So your direct repair mechanism can vary because the reaction is dependent on the damage being repaired. So we have 2 examples here. The first is DNA photolyase, which can also address thymine dimers. We already saw how nucleotide excision repair can fix thymine dimers, but we also have a direct repair mechanism for that. So this DNA photolyase uses a methyl tetrahydrofolate So that's methyl tetrahydrofollyl polyglucose, sorry. Okay. I mean polyglutamate. So methyl tetrahydrofollyl polyglutamate, which is considered a chromophore because it can absorb energy from light. So light of 300 to 500 nanometer with wavelength of 300 to 500 nanometer is absorbed by this chromophore in your photolyase. So this chromophore absorbs the energy and it passes the energy to a coenzyme FADH. Okay, so the FADH now passes on the energy to the pyrimidine dimer, and then the energy is used to create a radical. So it breaks the pi bond here. Okay, and then a series of radical reactions take place that removes or destroys the cyclobutane structure. And then once the dimer is fixed, the electron is transferred back to the coenzyme FADH. And then the photolyase has finished its job here. So it's ready for another round of direct repair once it has absorbed light energy again. Next is your O6 methylguanine methyltransferase. So this addresses the methylation of your guanine residue. So this methyltransferase, okay, uses a cysteine residue in its active site. So the methyl in your methylated guanine is removed. So the methyl there is removed and is passed on to this cysteine residue. Okay, so that you recover the original guanine nitrogenous base. The thing is, this methyltransferase is not exactly an enzyme in the strictest sense of the word, although it catalyzes the transfer of the methyl from guanine to the cysteine residue. Once you have the cysteine residue attached to the once you have the methyl residue attached to the cysteine, the enzyme is no longer active. So it cannot go for another round of catalysis. Okay, so it's a one-time enzyme. So that is direct repair. And that's it for DNA damage and repair mechanisms. So now we will be practicing translation, but this time in the context of DNA damage or DNA mutation. So we are given the original DNA sequence here, and this will be our template strand. So although it's not written, we will assign the left side of this strand as the 3' and the right side as the 5'. And we will be looking at the mRNA sequence. So that should be starting from the 5'. So we start with 5', T pairs with A, A pairs with U, C pairs with G, A pairs with U, C pairs with G, another G, A, C, C, G, C, U, G, C, U, G, A, and 3'. So this is our mRNA sequence. And then using our codon wheel, we're going to write the amino acid sequence from the mRNA transcript. So we start with, of course, the N terminal. the first, I forgot to mark the triplets. So AUG, UGG, AAC, CGC, UGC, and UGA. So now let's write the amino acid sequence of our uh, mRNA transcript. So of course, the first is methionine, and then we have UGG, which is a tryptophan, and then we have AAC, which is an asparagine, and then we have CGC, which is an arginine, and UGC, that's a cysteine, and of course we have stop codon. So that's the C terminal. So the C terminal residue is cysteine. Okay. Now, what if our DNA sequence is mutated? So we can actually identify. So let's see if we can identify the mutation. Okay. So what do you notice? Here, in the fifth position, instead of a C, we get a T. Okay, correct. So, what type of mutation is this? What's our answer? So, from a C, in the original DNA sequence, it became a T. So, is this transition or transversion? This is a transition mutation, right? Because you have pyrimidine, right? Pyrimidine to pyrimidine transformation. So from a C to a T. So that's our answer. This is a transition mutation. But this is if we are looking at the base alteration. Okay. What if we are asked what's the effect of the phenotype? So what type of mutation took place? So we have to look at the amino acid sequence. So let us write the mRNA sequence again, 5 prime. So for that, we have A, U, G, U, but this time it's an A, and then G, and then the rest are the same, AAC, CGC, UGC, and UGA. So this is our mRNA transcript. So let's look at the triplets. So AUG, UAG, AAC, CGC, UGC, and UGA. So what is the amino acid sequence? So from the N terminal, again, we start with methionine, followed by UAG. UAG is a stop codon. Okay, so what is the effect of this transition mutation? The effect is a premature stop. Okay, so we have a shortened gene product. If we have a shortened gene product, then what type of mutation would that be? So that is what we call a nonsense mutation. So this is a nonsense mutation if we are talking about the phenotype. Okay, I have another example here. So let's look at this DNA sequence, which is also mutated. And at first glance, you should be able to recognize that this DNA sequence is longer by one nucleotide, which means there should be an insertion, right? Can you identify at which position a nucleotide was inserted? So it's at the fourth position. A G was inserted. So the rest of your nucleotide is the same: ACC, TTG, GCG, ACG, and ACT. So you have an insertion there, which means our mRNA transcript would be also one nucleotide longer. Okay, so let's start with T pairing with A, and then A is with U, and of course C is with G. G is C, A is with U, C is with G, and then another G again, two A's, two C, one G, one C, one U, one G, one C, one U, one one G, and one A. That's the 3'. So this is our mRNA transcript. And then we're going to identify the triplets. Okay, since there is an additional nucleotide, we have here an incomplete codon. So it's just A. Okay, so what is our amino acid sequence? So again, from the N terminal, that's methionine. Then we have CUG, which is a leucine, followed by GAA, which is a glutamate, CCG, which is a proline, CUG, which is another leucine, and another CUG, and then there is no stop codon. Okay, so in this case, if this is just your mRNA transcript and there's no stop codon, then since there's no tRNA to enter the A site, then everything, your whole ribosome and other molecules involved in the process will just disassemble. Okay, so you get a different gene product in this case. So the effect is you have a different protein molecule, and then the mutation, we can identify an insertion mutation. And in terms of the phenotype, we get a missense mutation because we get a different amino acid starting from the second amino acid. Okay. And then I think I have another example here. For the last example, we have the context of sickle cell anemia. So you know the difference between a healthy person and one who is afflicted with a sickle cell anemia. So the mutation is in the gene that codes for the hemoglobin. So the oxygen transport of patients with sickle cell anemia is affected. And interestingly, the mutation happens only on one amino acid. Okay, so let's look at a segment of the normal hemoglobin DNA. So we're going to look at the mutation. So let's write the mRNA transcript first. So again, this is a segment of the hemoglobin DNA. So we have G, U, G, and then C, A, U, C, U, G, A, C, U, C, C, U, G, A, G, and so on. So this is just a segment of the mRNA transcript. And then let's look at the amino acid sequence. So for the amino acid sequence, GUG, GUG is valine, followed by CAU, CAU is histidine, followed by CUG, CUG is leucine, ACA is threonine, CCU is proline, and GAG is glutamate. So this is the normal amino acid sequence for this segment of the mRNA transcript. So now let's look at the sickle cell hemoglobin DNA. So can we identify the mutation there? So you see the T here, which is replaced by an A. Okay, so it's a transversion, right? Because from a pyrimidine, you get a purine in the deceased DNA. So let's look at the mRNA sequence. So from 5 prime, should be the same at first. So that's G, U, G, then C, A, U, and then C, U, G, A, C, U, C, 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 U, G, U, G. And I just realized there is a mistake here. So that's C, AU, CG, CG, TA, AU, GC, AU, CG, TA, GC. This should be U, right? So this should be U. That's CCUGA. So that's GUG. Let me just fix this. GUG, CAU, CUG, ACU, CCU, and GAG. So the fourth codon is ACU. ACU, which is still threonine. Okay, so we only got a mistake in the mRNA transcript, but not in the amino acid sequence. Okay, so this still translates to threonine. Okay, so let's go back to the hemoglobin, sickle cell hemoglobin. So let's look at the amino acid sequence. So again, identifying the triplets, GUG, CAU, CUG, ACU, CCU, and then GU. This would be G. This is GUG. Okay. So what is our amino acid sequence? GUG is still valine. Then we still have histidine for CAU. CUG is leucine. And then after leucine, ACU, which is threonine, CCU, which is proline, and then GUG. So it's not GAG, but it's GUG, which is a valine. Okay, so from a glutamate, so the thing is, from a glutamate here, which is an acidic amino acid, so it has a side chain which is negatively charged at pH 7. So its characteristic at this position, the position of your the characteristic of your amino acid at this position is now changed because you instead get a nonpolar amino acid, valine. So the reason why the hemoglobin becomes sickle shaped is that the red blood cell becomes sickle-shaped is because due to this single amino acid substitution or change, your hemoglobin molecules tend to aggregate with each other instead of retaining their native state. And this is driven by the hydrophobic interaction of these valine residues among the hemoglobin chains. That is why your red blood cells tend to form this sickle cell shape, hence the name sickle cell anemia. Okay, so what type of mutation is this? This is what we call a missense mutation because we get a different amino acid. Or in the context of the transformation of the nitrogenous base, this is a transition mutation.